Hi everybody and welcome to the Parkinson's Disease Genetics for Non-Geneticists course from the Aligning Science Across Parkinson's and the Global Parkinson's Genetics Programme. My name's John Hardy. I'm at University College London and it's my pleasure today to talk to you about the history of Parkinson's disease genetics. I guess I've been asked to give this talk because I've been around for so long I can remember some of the very early days of Parkinson's genetics. But it's been an interesting story, and I think that history teaches us quite a few things, not only about Parkinson's disease, but also about progress, how progress is made in science, and so it is quite a fascinating story. This will be the first module of 13 from our course syllabus. Here's a brief summary of my presentation. As a field, we have certainly come a long way. Over the last 20 years, in many ways, genetics has been the engine that has pushed us along on our voyage to understand Parkinson's disease. During this talk, it is not my intent to list each gene and locus, but to summarise key genetic advances done in the past and what the present and future in our field holds. I will start talking about how historically Parkinson's disease was considered to be a non-genetic disease and how major advances, including the discovery of point and gene dosage mutations in the synuclein gene, truly changed our field. Then I will tell you how the term autosomal recessive juvenile Parkinsonism arose and some curiosities about the discovery of the Parkin gene. In the late 1990s through to 2005, gene hunting was extremely competitive and fast-paced world, and I would like to show you how close these races to new mutations could get by talking about the discovery of LRRK2 mutations by two independent groups of researchers. I will also give an overview of what genetics has revealed thus far in the context of disease biology. Finally, I will briefly discuss how the GWAS era led to increased collaboration that allowed to the identification of new genetic loci for disease. These collaborative groups form the basis of large international consortium that now serve as discovery engines and large reference data sets in PD genetics. In this context, the Global Parkinson's Disease Genetics Programme will be crucial to promote open science and connect researchers worldwide to help unmask the contribution of currently uncaptured genetic variation to the risk of Parkinson's. So let's get started. One of the major reasons that Parkinson's disease's history is interesting is because for many years, in fact certainly until the mid-1990s, scientists, in inverted commas, knew Parkinson's disease was not a genetic condition. This is a strange phenomenon really, but I think it was mainly because most cases of Parkinson's disease did not have a clear family history, and I think there was an idea that what must be true for some Parkinson's disease cases had to be true for all Parkinson's disease. I would like to exemplify it with this study, which is actually from 1999, after the first gene for Parkinson's disease was found, that I will later explain. This epidemiological study of Parkinson's disease in twins kind of summarises that view. These were male twins from the army who had been in national service, and the authors came to the conclusion that Parkinson's disease, certainly over the age of 50, had no genetic influence. However, they did say that they thought that genetic factors might somehow be involved when the gene appears at or below the age of 60. At or below the age of 50. So there was very strong belief that Parkinson's disease was not genetic, very strongly. I'll just point out, though, there was a follow-up of this same set of twins much more recently by many of the same authors, and they showed that, in fact, the pairwise concordance for Parkinson's disease was about 0.2 and that heritability that was about 0.027. In other words, quite a considerable genetic influence. And the difference between the two studies that I just showed was that in this follow-up study, they had followed the twins for a further 20 years, and many of the cases, or some of the cases who were discordant in the previous study, were not discordant in the later study. In other words, in the intervening 20 years, they had developed disease, and I think that really illustrates the problems that epidemiologists have with genetics. I'm just going to remind you to look at this heritability here of 0.27 in the later study, because what we now know is that the epidemiologist's view of things has moved towards the same similar heritability to the GWAS work in more recent studies. And you will learn more about the concept of heritability in the next module by Dr. Zivgan Orr. And in fact, to jump a long way forward, genetic analyses performed by the International Parkinson's Disease Genetic Consortium really gives the same heritability estimates using molecular DNA data. So in fact, there is now concurrence between the moderate heritability of Parkinson's disease overall, both from molecular genetics and from epidemiological data. What I would say, just to jump forward to that, is that this heritability of about 0.27, we have found molecular data to explain about 30% of it. In this regard, you will learn more about genome-wide association studies and how this has been estimated in Module 6 of this course from Mike Knowles and Mary Macarius. Now, given that, it's worth just shouting out a couple of clinicians who really pioneered the study of genetics of Parkinson's disease as a result of intuition and hard work. Roger Duvoisin and Manfred Munter. Roger Duvoisin was in New Jersey and Manfred Munter was at the Mayo Clinic. And they both collected very large pedigrees with clearly autosomal dominant patterns of inheritance in which an affected individual has one copy of a mutant gene and one normal copy, as you will see in a module 2 of this course. I'll show you the pedigrees in the next couple of slides. And the other shout out, so to speak, is to Japan, where clinicians had long recognized a recessive disease which they called autosomal recessive juvenile Parkinsonism. Note, not Parkinson's disease. So they had identified these recessive, often consanguineous marriages which led to children with Parkinsonism. So this is the family that Roger Duvoisin characterized, the so-called Conterzi kindreds, originating from a small city in southern Italy. As we can see in this slide, a total of 41 persons in 4 generations have been affected with Parkinson's disease, with an early onset of about 45 years and a rapid disease course. And this study was published in 1990. And in fact, I was at an NIH study section, though not the reviewer, which reviewed a grant application by Duvoisin for his analysis of the Conterzi kindred. At the review section, this grant was triaged, which means it wasn't reviewed, because the primary reviewer said, "We know that Parkinson's disease is not hereditary, so why is this group deciding to study the molecular genetics of Parkinson's disease?" And they said it would be much more valuable if they looked at the well water and agricultural practices for the individuals in this pedigree, because obviously they felt there was something in the environment which was causing them to get Parkinson's disease. So really, it was quite a remarkable scientific blindness to study the genetics of the disease. Fortunately, really, the NIH and the NINDS realised that mistakes were being made by the review committees and that these were likely to be genetic problems. And the NINDS sought the aid of Bob Nussbaum and said, please give him all the DNA samples and let's see if we can find the gene in this Conterzi kindred. And so Roger Duvoisin gave Bob Nussbaum at NINDS and his then postdoc Mihail Polymeropoulos the samples, and almost immediately they mapped the gene for Parkinson's disease to chromosome 4p. They then sequenced the genes in that linkage area and found mutations in the synuclein gene, encoding the synuclein protein. This slide therefore represents a major discovery for Parkinson's disease genetics. As geneticists, the late 1990s and early 2000s were an exciting time when the major accomplishments centered almost completely on monogenic forms of disease. And this is the family. The family was from southern Italy in this area here. This is the Contursi area just outside Salerno, but actually the vast majority of cases with this mutation were subsequently found in Greece. And this really harks back to the history of the Greek Empire and then the Roman Empire, and when this was really one political entity at the time, and the majority of cases with this first mutation are in fact of Greek origin, and it accounts for a reasonably high proportion of Greek cases. Later on, pathologists and histopathologists such as Maria Spallanzini used synuclein antibodies to study the protein and showed that synuclein was the major histopathological hallmark of Lewy bodies. And really, that was the icing on the cake, and showed that Polymeropoulos' and Nussbaum's paper really was opening the way forward for the disease. It provided empirical evidence that there was a genetic basis for rare forms of the disease, and because alpha-synuclein was a major component of all Lewy bodies, that these findings were directly relevant to all cases of PD. And subsequently to that, we and others have now found a whole range of different point mutations in the synuclein gene that you will study later in this course as part of Module 3. And what you see here is the structure of the synuclein protein and the places so far at which mutations have been identified. I'll just say something about the structure. We don't fully understand it, and there's a lot of argument about the precise structure of the protein. But what I think is going to be important is that it's a membrane-associated protein and it's very highly expressed. I will now talk about Manfred Munter's pedigree, published in 1998 before Polymeropoulos and Nussbaum's discovery. Although it had actually been published much earlier by George Spellman in the 1970s, I'll just point out this title. As a curiosity, the Annals of Neurology journal would not allow this to be called Parkinson's disease because, as I've said, everybody knew that Parkinson's disease was not hereditary. So you can see that full knowledge, if you like, false knowledge, if you like, was an inhibitor of progress. The kindred that Manfred Munter started to collect at the time was continued by Katrina Gwynn. This is what was called the Munter's kindred. Katrina Gwynn and one of the families linked this to a California kindred, and this led to the Iowa kindred. Then, in 2003, Andy Singleton showed the same synuclein haplotype among the affected individuals, although we couldn't find an amino acid change because the genetic cause was not a missense mutation. And using gene chip analysis, Andy showed there was a change in the gene dosage. Here you can see in blue the dosage along chromosome 4, and we observe at some point there is a change. What we observe here is that there was a triplication of the synuclein gene, and you can see that beautifully here in the in situ. At the beginning there is a normal chromosome 4 with one copy of the synuclein, and on this peak here is a mutated chromosome with 3 copies. This shows the triplication very clearly, and this beautiful work was done in Bob Nussbaum's lab. Just also, I should say that in the majority of cases, the age of onset in this family is in the 30s. Most family members start with Parkinson's disease and they go cognitively off, maybe 5, 6 or 7 years after their movement disorders problems started, but some individuals, and this is one of them, go off very quickly with dementia before they develop, if they ever indeed develop, movement disorders. So they clearly have dementia with Lewy bodies. For instance, in this example below, the affected individuals is doing a clock drawing test in 2000, and then only a couple of years later the person is finding it very, very difficult. Later in 2003, they can no longer do the clock drawing test. This person had very mild movement disorders by this time, and so clearly in this person dementia is the dominant part of the phenotype. And I think it's very interesting to look at the videos from this family, which Katrina Gwynn has put online in movement disorders. This is the reference for it, and I would really suggest looking at the videos of the pedigree, which you can find at the reference. And just to make a separate point, and that is that we, of course, are looking for mechanistic therapies for Parkinson's disease. That's why we're trying to find genes after all. But it's worth saying that L-DOPA makes a huge difference in this family. So all this family has the same cause of disease. They've all got a synuclein triplication. And what you can see here is that those family members who lived before the era of L-DOPA are basically all dead within 7 years of diagnosis, whereas those in the era of L-DOPA therapy were still living. In fact, it seems as if the median age of death now seems to be about 17 years after the onset of the disease. So really, I think that L-DOPA has really changed the progression of the disease as well as being a symptomatic therapy. And I think that that's something we mechanistic geneticists need to take on board. As I briefly outlined at the beginning of the talk, in Japan, the Japanese clinicians performed this study in 1996, and there are earlier studies as well looking at these families with autosomal recessive disease. They were usually consanguineous marriages where the age of onset was between 15 and 20 years typically. And so these were the families they looked at, but you'll notice they're careful not to call it Parkinson's disease. And you'll notice here in the last sentence of the abstract, the similarity of clinical findings in these patients and the differences from other types of Parkinsonism indicates this is a distinct clinical entity. In other words, not Parkinson's disease in their view at that time. As you can see, this is the sort of family they were looking at, autosomal recessive disease, as you will further learn in the next module. This means that the parents are not affected by Parkinson's disease, but they carry a single disease-carrying variant in the heterozygous state. When one of the offspring inherits the pathogenic allele from each of the parents and therefore carries a variant in a homozygous state, the disease appears. And as a result, they found this gene, which they called the Parkin gene. Well, why did they call it the Parkinson gene? Here also there's a kind of dirty little secret, in that they realised if they were to get their paper into Nature, they couldn't say they'd found a gene for a rare Japanese disorder, because that would get into a decent genetics journal, but it would not get into Nature. And so they named the gene Parkin and started to elide the difference between this disorder, autosomal recessive juvenile Parkinsonism, and Parkinson's disease. And how they found it was by performing homozygosity mapping. I'll show you an example of that later. And what they identified was exon deletions, which are actually the easiest type of mutations when you do this sort of analysis. So that's how they found the gene. And you will learn more about copy number variation later in this course. And I should say that once Parkin mutations were found, all the objections to calling ARJP Parkinson's disease also disappeared. So that's really showing in a way how science operates. I will say that, there is an argument on both sides. We did a study of the Parkin cases in the UK by exploring 6 brains here in our brain bank. So 4 are from the UK and 2 from Southern Ireland. And interestingly, they had no Lewy bodies. So the question of whether it's a different disease or not comes essentially down to a semantic argument. Continuing with the history of Parkinson's disease, I'll also talk about the identification of LRRK2 by two different research teams, which was a major discovery in the field. The first was a collaboration between the NIH group, which Andy Singleton led, and others, and a Spanish group with Jordi Perez-Tur and Nick Wood's group, who is here at Queen Square. The second group was led by Tom Gasser and Matt Farah. These 4 families came from the Basque region of Spain. A linkage to chromosome 12 had been identified and called PARK8 by Japanese clinicians, and Andy started to study polymorphisms on chromosome 12 in these 4 families from the Basque region, which is a geographic isolate in northern Spain. And the reason I show this is because really it's a beautiful use of the technique of positional cloning. These families showed linkage to chromosome 12, but better, what they showed was that all the individuals on chromosome 12 in 3 of the families showed a very large area of haplotype sharing, but in 1 of the families they only shared a small part of the region shown here, and this reduced the area that Andy thought the gene was from about 70 genes to about 12, and clearly that reduced the amount of sequencing he had to do. And this really was the key to the identification of LRRK2 in 2004 as a cause of autosomal dominant Parkinson's disease, and the most common monogenic form of Parkinson's disease identified till date. This was exciting for him and for our lab generally, because we were in a good race against a good friend and colleague, Tom Gasser and Matt Farah, and this has enabled us, let's say, to draw the race. The 2 papers identifying LRRK2 as a PD gene appear back to back in the same issue of Neuron. The other thing I'll mention is, as I show this slide, is that both papers identified LRRK2 and found a few mutations, but I'll also do a shout out for this paper from Tob, Zig Zolik and Matt Farah and their colleagues, because Zig had collected these families and not all of them had Lewy body pathology. Some of them had tangle pathology, and our brain bank here at Queen Square, we have 7 LRRK2 mutation carriers, which is common in Northern European population, of which 6 have Lewy bodies and 1 of them has tangle pathology. So that's something I think we want to explain in the long run. So I've already talked about the cloning of Parkin. Also, by rather similar methods, other recessive genes were found. PINK1 by Nick Wood and Enza Maria Valenti, DJ1 by Peter Hertig and Vincenzo Bonifato, and FBOX07, which was identified by Elahi in Iran. I would like to mention that current technologies, including whole exome sequencing and whole genome sequencing, did not exist at that time, and those now afford a much more complete and much more rapid detection of genetic causes of familial forms of disease. This type of data is now being generated in a large-scale manner and easily shared across sites, particularly using cloud-based infrastructure. Such sharing not only increases the power to detect genetic variants of interest, but also serves to produce a reference PD dataset that the field as a whole can use. Of note, I would like to talk about GBA, one, if not the most relevant genetic risk factor for Parkinson's disease. And here it's worth giving a shout out to Ellen Sedransky and her team. It was known that homozygous GBA mutations gave rise to Gaucher's disease, the most common lysosome storage disorder, The finding that heterozygous GBA mutations predisposed to PD was a major discovery in our field and was made through keen observation by Ellen and her colleagues, who noticed that the parents and grandparents of children with Gaucher's disease had increased incidence of PD by about fourfold. While this idea took time to be proven and accepted, it turned out to be a critical observation. So it was really not through detailed molecular genetic analysis, but largely through astute clinical observation that GBA was identified. And interestingly, this find has been expanded to include other lysosomal storage disease genes, particularly through the work of Josh Schulman and Peter Hertink and colleagues. So to jump now, and really not to go over the cloning of all the other genes linked to Park loci which have been identified, I'm going to go through them very briefly so that you can get familiar with them and the biology behind them. Synuclein, which I've widely talked about, is a membrane protein and it's somehow involved in membrane fusion. These genes, these recessive genes, which you'll see later in the course, including Parkin, PINK1 and DJ1, are involved in mitophagy. And mitophagy is the process by which damaged mitochondria are removed from cells. GBA and these other genes are all involved in the endosome-lysosome pathway, so they take part in the process by which lysosomes degrade cellular components. So really I think this points to the future. We are beginning to see now that the molecular genetics is not just identifying genes, but it tells us which cellular processes are going wrong. I'm now going to move on and talk about the GWAS for the disease. What I've been talking so far is Mendelian disease, where you have clear modes of inheritance. But of course, the majority of cases of Parkinson's disease, as I've said, don't have clear modes of inheritance. And starting in the early 2000s, there started to be a whole series of analysis, first led by Tom Gasser and Andy Singleton, and then joined by all the rest of us in the IPDGC, looking at GWAS for Parkinson's disease. That's looking for genes which don't cause disease, but regions of the genome which slightly individually increase your risk of disease. And I just show this slide to illustrate how the number of cases increases with time as more and more samples are collected, and also to make the point, and one of the things that we have is really correct, is that nearly all the study are Europeans, and obviously that's not satisfactory. We need to understand the genetic and architecture of Parkinson's disease in Asian populations, Latino populations, and in Africa. And as you will see in the module Understanding the Genetics in Underrepresented Populations later in the course by Nacho Mata, this clearly has to be a major goal for the future, and GP2 is going to be key to achieving it. We still only know a fraction of the total hereditability of Parkinson's disease, some of it presumably still hidden in the huge number of rare coding variants that are to be found by whole exome sequencing. And these data are going to be fully interpretable only when much larger cohorts are available that allow us to look at genetic variability at much higher resolution than to date. This is one of the early studies. I'll just make a little point. This is synuclein here. So this is saying not only is synuclein a Mendelian cause of disease, it's a risk locus for the disease. This is the tau haplotype. I'll just say now that we think that it is not tau which is important in this locus, but a gene called CANSL1. And my colleague, Dr. Helen Plum-Favreau, has got beautiful data showing how much progress has been made to define this risk gene. She has a paper on bioRxiv at the moment. And in this study, as many of the GWAS have been led by Mike Knowles, I won't go into detail about the gene of GWAS because you'll learn about it later. But the point I'll say is that many of the candidate genes in these risk loci are lysosomal or mitochondrial. So fitting for the Mendelian, so fitting really with our understanding of the Mendelian genes. This just makes the point explicitly: lysosome, mitochondrial, immune and dopamine metabolism genes are involved in the pathogenesis metabolism. I'll just add that dopamine metabolism happens largely in the mitochondria, and so it's probably part, if you like, of the mitochondrial issue. Well, how to put this together? I'm not going to really go from a history. My hypothesis, and I'm sure other people have had similar ideas, is to try and put this together. This is, I think, a very important paper from Dimitri Krank, and he showed two things. Synuclein is metabolized through the lysosome, and increased expression of synuclein inhibits lysosome flux. So I think it's an important point which starts to draw important these important findings together. The important point, not strictly a genetic point, is this beautiful paper showing the electron microscopy of Lewy bodies. And the reason this is a beautiful paper is because, as I've said, we know synuclein is the most prevalent protein in Lewy bodies because of Murray Spilantini's work. But what this paper shows here is that there's lots of other components, membranous components, and remember that I said synuclein was a membrane bound protein, and most especially mitochondria. So I said mitophagy is the process by which mitophagy are sent, if you like, to the lysosome degradation, and here you can see the lysosome, the Lewy body rather, is packed full of not only of synuclein, but also of mitochondria, and I think that starts to allow us to bring the whole thing together. And here is really my final data slide before the summary, and this is a slide that my colleague Patrick Lewis made maybe 10 years ago, saying that these two processes were important, and I think this very accurately summarizes my current view. We have two processes which are a problem in Parkinson's disease: mitophagy, which are a process by which mitochondria are sent to lysosomes, and the autophagy process. And as I have said, autophagy is often a process by which membranes are cleared through the lysosome, and synuclein is attached to the membrane, and I would regard these as competitive processes. So remember, this is a hypothesis, not a fact. So I'm suggesting that lysosomes are degrading membranous material which contain synuclein, and they're also degrading mitochondria, and these two, if you like, are competing for lysosome space. And I would say what's happening in Parkinson's disease is that you're overloading this clearance system. And what you're looking at is the clearance of damaged mitochondria and synuclein on damaged membranes. I would also suggest the mitophagy process is largely a dopamine process. Only Parkin, PINK1 and DJ1 don't extend beyond the nigra, whereas autophagy process happens in all neurons. And so I would say the more mitophagy is wrong in an individual, the more benign the disease is, because the more likely it is to stay confined to the nigra. But I emphasize that this is a hypothesis and not a fact. So just to summarize my key messages. Until 2 decades ago, Parkinson's disease was considered the archetypal example of a non-genetic disease. The identification of gene-causing mutations in synuclein marked the starting point for Parkinson's disease genetics. The identification of rare recessive forms of disease associated with early onset disease caused by Parkin, PINK1 and DJ1 supported the notion that PD was to some extent genetic. In the GWAS era, great advances have been made in identifying many risk loci for disease, and genetics is now key in informing biological pathways involved in the disease. etiology. And increasingly, this success has been driven by extensive scientific collaboration, and future collaboration will not progress without teamwork in an open scientific environment. Thanks very much for listening. I hope you found this module interesting, informative, and possibly entertaining. Thank you very much indeed.